Okay, so we're going to take a look at chapter number 20. We're just going to look at the first half of chapter number 20. We will stop when we get to the brain because that content we will cover together before we do our brain dissection. So looking at the first part of this chapter, we're going to talk about coverings of the brain and spinal cord. So with the brain and spinal cord, these two components of your central nervous system, it is imperative that we have very well protective coverings for them. So our first covering is our outermost covering, and it is a hardened covering, so we're looking at bone. Okay, so this bone is going to encase the brain and the spinal cord. So of course we have our cranial bones that we studied with the skeletal unit. So you have your parietal, occipital, frontal bones, temporal bones, and then of course the bones that you find on the underside of the brain as well. Okay, so these cranial bones are going to completely encase the brain and protect it. We also have our vertebrae, which are going to kind of form a bone-like ring around the spinal cord and encase and protect the spinal cord. So not only do we have bone, we also have an inner covering. And the inner covering we refer to as the meninges. And with the meninges, we do have three different types of meninges that we will talk about, but these are going to be a membranous type of covering, so a membrane tissue type of covering. Okay, so we have the bony covering, and then we have the meninges, which the meninges are deep to the bone. We see this picture right here. So up here we can see the brain. Of course, you can't see the bony encasement or the meninges associated with the brain. With this picture, otherwise we will not be able to see the brain. We can see different parts and components of the brain that we will talk about prior to doing a brain dissection. But as we move on down to the spinal cord, a couple of things that we notice. So your spinal cord runs down to about your first lumbar vertebrae. That's where your spinal cord is going to end distally. Right before it ends, you notice that there is an enlargement, and we call it the lumbar enlargement of the spinal cord. So it's pretty large at the end. And then there's also what we refer to as the cervical enlargement of the spinal cord. So it's a little bit wider in these places because in these places it's going to lead to what we call a plexus. Okay, so we'll see what this plexus looks like a little bit later, but it's a network of nerves, so a network of these spinal nerves that are going to leave or come to the spinal cord. So there's a big cluster of them in the cervical region and then a big cluster of them down here in the lumbar region. But on this picture you can see the bony covering, the encasement around the spinal cord, which would be the vertebrae, of course, and then a membranous covering, which would be the meninges themselves. So let's explore the meninges a little bit. So with the meninges, there are three layers. Here we can see the names of the three layers. We have Dura Mater. Deep to that, we have Arachnoid Mater, and then deep to that, we have Pia Mater. Okay, so this picture, I just grabbed this one off the internet, but here you can see cranial bone of the skull. So you're looking at like parietal, for example. But deep to that, you find the Dura Mater in blue, and then Arachnoid Mater in red. There's a little bit of space right here in purple, and you'll notice it's called Subarachnoid Space. Sub means deep, so deep to the Arachnoid Mater, or beneath it, we have some space with that fluid that we talked about last class, okay, last lecture, which was CSF, cerebrospinal fluid, sitting right here. And then in yellow, we have our third and deepest layer, which is our Pia Mater. Pia Mater has these blood vessels associated with it, as you can see with this picture. And then we have the gray matter and white matter of the brain. Okay, so that's kind of the different layers and their locations in relationship to each other. This picture right here shows all three layers yet again, but this time we're looking at the spinal cord. Again, Dura Mater, with this picture is in purple, and we can see it's the outermost. And then as we go a little bit deeper, we have the Arachnoid Mater here in pink. And then as we go a little bit deeper, kind of this peach color right here, really thin, we see the pia mater, and then this is just the spinal cord. Okay, so again, most superficial is dura, then arachnoid, and then deepest, attached directly to the spinal cord and directly to the brain, is your pia mater. So let's just kind of explore each of the three. So Dura Mater, again, is super strong. It is your white, very dense, fibrous tissue. Very strong, very tough to cut through. When we do our brain dissection and we have some Dura Mater associated with it, it'll be pretty difficult to get your scalpel to slice it. Okay, so we can see it is the outermost layer of the meninges, and it makes up the inner periosteum, so the inside of the cranial bones is where we find that Dura Mater. So sometimes you lose some of the Dura Mater when you remove the cranial bones because it is attached to the inside lining of those cranial bones. So the periosteum is what surrounds the bone. This is on the inside of them, of course. And again, Dura Mater, first layer that we have right here. Next up, we'll talk about the Arachnoid Mater, which we can see here in pink. In arachnoid, you should probably be thinking like spider-like, spiderweb-like, because arachnoid refers to spider. So it does have that cobweb appearance to it. Okay, it's very delicate. So oftentimes you don't really have it when you do the brain dissection because it's so delicate and it's hard to see. A lot of the cobwebby pieces, they're called trabeculae because they kind of extend like the trabeculae of spongy bone, but they fall down and get all messed up when we actually access the brain. Okay, so it's very delicate and cobwebby-like, and that's your middle layer between the dura and the pia. So we hop back to this picture. Here's your arachnoid in red between the dura in blue and the pia right here. So speaking of the pia mater, that's going to be the deepest one. So innermost transparent layer. Transparent in the sense that it literally is going to look like saran wrap, plastic wrap, stuck to the brain. Okay, you might not even realize it's there until you try to like twist or move the brain a little bit and you'll see it wrinkle up. So it is physically adhering to the outermost surface of the brain. And we do find blood vessels. BVs just stands for blood vessels. Okay, so it does physically attach to the outermost region of the brain and the spinal cord. So here we can see the skin, okay, and then your bone, the outer periosteum, but the inner periosteum with the cranial bones is your Dura Mater. And then your Arachnoid Mater, which is very delicate, that if you take the bone off, sometimes the Dura Mater would go with it, of course, and then the arachnoid gets messed up. So really all you're left with is the pia, which is stuck directly to the brain. It is physically attached to the brain. That one's going to be really difficult to remove. You can probably just kind of pick off little pieces, but it is stuck directly to the brain, and it follows in those like grooves and ridges associated with the brain. And here we can see it in yellow, and you can see the blood vessels. The red and blue squigglies, those are your blood vessels. This picture right here, we're looking at a dissection of the spinal cord. So we can see we're looking at the distal region of the spinal cord. So we can see how the spinal cord ends. So here you can see the very thickened lumbar enlargement, and then it ends down in that lumbar region, just at the start of the lumbar vertebrae or so. And then we can see the Dura Mater is cut, so we can have access to the spinal cord itself. Conus medullaris, that's the distal end of the spinal cord, where it comes to kind of a cone-like tip. So you can see it kind of comes to a point right here. That is your conus medullaris. Okay, so the cone-like tip point of the spinal cord. It's kind of right here on this picture. You'll be asked to label this in one of your labs, which is due. And then we can see a lot of fraying of these nerve roots. You can see all these little pieces where it kind of looks like a ponytail. So they call it the cauda equina. Cauda means tail, equina means horse. So horse's tail or ponytail. A bunch of these little strands together, like a ponytail. And then you have little pieces that kind of blend in with the coccyx, okay, and that's called the filum terminale. Okay, so the terminal end of everything associated with the spinal cord. So a little bit of pia mater kind of blending in with the coccyx. Here we can kind of see another look at the transverse type of a cut. And if we look at this picture, we can see a few things. First off, in purple, you can see the Dura Mater, okay? And then you can see right here in pink is your Arachnoid Mater, okay? And then in peach, right here, it's kind of hard to see with the contrast, but this is your pia mater. And then we have some space. We have epidural space. Epidural space you have with the spinal cord. That's going to be the space between the bone of the vertebrae and the Dura Mater. Remember, it's impossible to have this space with the brain because the Dura Mater is stuck directly to that cranial bone. Remember, the Dura Mater is that periosteum of the bone. So you don't have epidural space. This is just filled with a lot of fat, but you don't have this epidural space with the skull. You only have it with the vertebrae. So that's something I wanted to point out with that picture. Okay, so a little bit more with these spaces. Okay, so epidural space is first. Epi means on top of. Dura means Dura Mater. So on top of the Dura Mater, so between the bone and the Dura Mater, you have the epidural space. You don't see it associated with the brain. You don't see it with the skull. You only see it with the spinal cord. Okay, so a lot of fat in there, other connective tissues. Subdural space, sub means beneath, so beneath the Dura Mater, but before the Arachnoid Mater, you have subdural space. Okay, there's some fluid in there. And then you have subarachnoid space, and this is the most important of the three. Subarachnoid space means beneath the Arachnoid Mater, so that'd be between the arachnoid and the pia mater, is a space filled with CSF. Your subarachnoid space is filled with cerebrospinal fluid. This is going to give your brain and your spinal cord a little bit of buoyancy. Okay, so it has that space within the bone to just kind of hang out. So it's that lubrication and buoyancy around the brain within the skull. So out of all these spaces, you absolutely must know the subarachnoid is a location, subarachnoid space is a location where you have CSF, cerebrospinal fluid. Okay, and then this picture right here does show the subarachnoid space with the CSF. Okay, so arachnoid mater's in red, here's pia in yellow, this is the space right here. So again, it's some cushioning for the brain to hang out within these meninges, and within the bones of the skull. Okay, so the CSF, cerebrospinal fluid. Again, it's going to help to support and protect. It's going to provide a nice cushioning. And like we saw in the last chapter, in chapter 18, in our nervous system cells chapter, this fluid isn't stationary. It is continuously circulating. We saw the ependymal cells with their cilia are what's going to keep them circulating. Purpose of the fluid is to constantly be circulating to provide some support and protection, yes, but then also we have a lot of receptors located all along where we find the CSF, and they're there to detect if there's any changes with what is found within the CSF. Okay, so we can actually pull some of the CSF out, and the most common way that is done is going to be via a lumbar puncture, which is also known as a spinal tap. Okay, so we're going to reach in and from that subarachnoid space, take a little bit of the CSF, and then they're going to observe it and see if they find anything suspicious that they could link to a diagnosis. Okay, so we can monitor it, we can test it, and so on. So where exactly do we see it? Well, first space, you guys already know, or first location, you already know. That would be the subarachnoid space. Okay, so that's where we find CSF. Where else do we find it? So yes, subarachnoid space is around the brain and spinal cord. We also find it within the central canal of the spinal cord. That means if you look at the spinal cord, I do have a model. Here I can show you. We'll have pictures of it too. Let's just see how well this model works. Maybe I need to go a little bit closer. Let's see. But right here, right smack in the center of the spinal cord, hopefully you can notice like a little hole right here. That little hole is a central canal. So running right smack through the center of the spinal cord, we find a lot of the CSF. Okay, so subarachnoid space, central canal, and then also we're going to find it within the ventricles of the brain. Ventricles are open spaces in the brain. I have a model right here. This model is showing, this is kind of a plastic model. All these gray components are open spaces within your brain. Okay, so these are the ventricles on this model. So I have some pictures I can show you, and then we'll reference that model yet again. So let's look at these pictures. First off, looking at this image, so here we can see this is going to be your subarachnoid space. Okay, so this is between the arachnoid mater and white. You can see the cobwebby appearance. And then the pia mater, okay, which is right here in white, which is stuck directly to the brain. So all in here surrounding the brain, between the bone and the brain, you're going to find CSF within the subarachnoid space. This is going to continue down around the vertebrae, or down around the spinal cord as well, within the vertebrae. Okay, so not only around the brain, but also around the spinal cord. This picture right here is pretty good in the sense that right here, if you follow this arrow, this is the central canal of the spinal cord, right through the middle. Here you can see it labeled central canal. So this is just going to be the middle, that pinhole running right through the middle of the spinal cord. Subarachnoid space is the space on the outside of the spinal cord. And we can see that space if you follow this purple arrow. The subarachnoid space, and we'll follow it all around. Here's subarachnoid space. It does follow all the way around the brain. And you can just kind of follow it. Okay, so it surrounds the entire brain. You can see it right here as well. And it surrounds the entire spinal cord. So subarachnoid space around the brain, subarachnoid space around the spinal cord, central canal within the spinal cord. What we didn't mention yet are these ventricles. So I'll show you another picture and point out the ventricles. Yeah, they're labeled on this picture. You can see lateral ventricle, third and fourth. There's actually 2 lateral ventricles. So 2 laterals plus a third and a fourth gives you 4 total. Okay, so this picture right here, anything in blue is a ventricle. So here we can see lateral ventricle are these big C-shaped structures. This is one lateral ventricle. There's another one right next to it. So your brain is split into 2 hemispheres. You have 2 sides of your brain. Each side of the brain has one of these lateral ventricles. So if I hold up my model, same orientation. Now that looks backwards. Everything's backwards with this video. But this big C-shaped right here, this is a lateral ventricle. If I hold it like this, you will notice that there's one here, and then one here. So there's 2 of them, one on each side of the brain. And actually, I believe I have a picture that shows that a little bit better. Yep, right here. So this entire thing is one lateral ventricle, and this is the other lateral ventricle. So one on each side. Oh, this is even better. So if you look at this picture, here's a lateral ventricle on the right side, here's a lateral ventricle on the left side of the brain. So 2 lateral ventricles. I'll just leave this picture up. It's kind of the best one. Here we see a third ventricle, which is right smack in between the two of them. We have something called an interventricular foramen that carries CSF from the lateral into the third. Okay, so interventricular means between the ventricles, there's an opening. Okay, so a little foramen, an opening between the lateral and the third. And then down here, this big diamond-shaped one, that's your fourth ventricle. And then there's a long passageway to it. This long passageway is called the cerebral aqueduct. Okay. And then we have lateral aperture and central canal, okay, which is going to allow us to have access, lateral aperture to the subarachnoid space and central canal right down the middle of the spinal cord. Make certain that's not so blurry. Let's try another picture. See if we can get that blurriness to go away. Let's try this. There we go. Okay. So if we look at this picture yet again, here we can see this is a lateral ventricle. There's two of them side by side. You can kind of see the second one back there. Here's your third ventricle. To get there, you're going to have this interventricular foramen, okay? And then you have this long cerebral aqueduct from the third down to the fourth, and then into the central canal of the spinal cord. So absolutely, you should be familiar with locations of these ventricles. When we do a brain dissection, I will have each group point out all four of these ventricles, so you need to be able to find them. And there'll be the open spaces. So how do we make the CSF? Remember in the last chapter, we talked about the ependymal cells. They looked like epithelial cells. They had cilia. So they're the ones that make the CSF, and they keep it moving around. Okay. Also remember from that lecture, I said that they can exist in packets. So a big clump of ependymal cells together, the ones that actually make the CSF, are called this. They're called choroid plexuses. So plexus refers to like a big group. So choroid plexuses are a big group of ependymal cells that will make the CSF. The CSF is very similar to blood plasma. Okay, so just pull it right out of the bloodstream, and it passes through the CSF, passes through the ventricles and subarachnoid space and central canal, and then it goes right back into the bloodstream. So it's just kind of like it's pulled out, travels through, goes back to the bloodstream continuously all the time. And this is your flow right here. So yes, I expect you guys to know the flow. I showed it to you on the other picture, but remember lateral ventricles through the interventricular foramen. Now you're in the third ventricle. You travel down that long cerebral aqueduct. Now you're in the fourth ventricle. Then you can go to the subarachnoid space and the central canal of the spinal cord. So if we look at this picture, this right here, this is the lateral ventricle. Okay, there is a choroid plexus present. That's just kind of this big, thick area right where my arrow is right now. There's also a choroid plexus of the third ventricle. That's kind of this big, thick, kind of pink area right here. So we have them in the lateral ventricle. We have them in the third ventricle. And then we also have a choroid plexus down here in the fourth ventricle. So within all four of your ventricles, you have a choroid plexus. So remember, the choroid plexus is what pulls the CSF out of the blood and allows it to travel through. Okay, so your choroid plexuses are what technically makes the CSF, and again, the CSF is just taken right from the blood. This picture right here, again, shows the flow. So here we can see lateral ventricle. Here's your little foramen. Okay, your interventricular foramen into the third ventricle, down the long cerebral aqueduct into the fourth ventricle. Okay, we can travel out to the subarachnoid space, or we can continue on right where the arrow is right now, and this is the central canal of the spinal cord. These purple regions are those choroid plexuses. There's also one up here in the lateral ventricle, not shown. So there's one in each lateral ventricle, also in the third, also here in the fourth ventricle. It only looks like half of a diamond shape because you're only looking at half the brain. If you're looking at the entire brain, it would take on a full diamond shape, of course. Okay, and we know that it travels in this subarachnoid space as well. Okay, subarachnoid space you can see is kind of this like light blue color. This says venous blood because eventually the CSF has to get back in the bloodstream. And we can see these little pouches are where the CSF goes back into venous blood. So it travels in the subarachnoid space, enters into one of these little pouches, and now is in this dark blue area, which is going to be the venous or sinus system of the bloodstream. I'll show you that in just another slide or two. Okay. So from the fourth ventricle, of course, we can go right into the central canal, as we mentioned, or we can go into the subarachnoid space. And it'll travel through that subarachnoid space like we just saw, or it's going to enter into one of these little pouches called the arachnoid villi. So arachnoid villi are like little bundles where the CSF will gain access to the venous blood supply. So if we zoom in right here, in pink, this is a subarachnoid space. This is an arachnoid villus. Multiple ones are called villi, that's plural. So we follow the red arrow, the CSF enters into these, and now it's in the blue region, which is your venous blood supply. So it's in a vein. Veins associated with the skull are called sinuses. So we can see that here. I'm just looking to see if it's labeled sinus anywhere in this picture. Yep, over here. Superior sagittal sinus is the name of that vein. So yet again, let's just do one more review. This is your lateral ventricle. It has a choroid plexus. Here's your third ventricle. It has a choroid plexus. Here's your fourth ventricle. It has a choroid plexus. Okay, so the CSF, again, gets made in these choroid plexuses, spills into, I'm just going to start at the lateral ventricle. You can really start at any ventricle, but I'll start lateral ventricle. Passes through this interventricular foramen. You can see that labeled right here. Enters into the third ventricle. Passes through this very long cerebral aqueduct. See it labeled right here. Comes on down to this fourth ventricle. And then we have two things that can happen. One, we can pass through this median foramen and enter into subarachnoid space. Okay, or lateral foramen, okay. Enter into, you can see another one right here, that's the lateral one. Okay, so you're gonna enter into one of these foramen, medial-lateral, and get access to the subarachnoid space, or CSF continues down the central canal of the spinal cord. But we can see this is that subarachnoid space all around the spinal cord and all around the brain. And it's gonna continue around that subarachnoid space until it hits these arachnoid villi, and then it'll enter these arachnoid villi and spill into the venous, oxygen poor, blood supply, such as the sagittal sinus, which is the vein, of course, running along the brain, as you can see right there. Here's a video where we can see flow of CSF fluid. Pretty good video. So first off, we can see the ventricles. You can see the horn-shaped lateral ventricles, kind of like the horns on a ram. In the middle of the C shape, you can see that third ventricle. And then, of course, down here you can see the fourth. And And then, of course, this is that interventricular foramen. So from the lateral ventricle through that interventricular foramen to the third, down the long cerebral aqueduct, down here to the fourth, and then we can go subarachnoid space or central canal. You see venous blood supply up here, venous blood returning to the heart. So venous blood supply, so superior sagittal sinus is up here. CSF is produced at the ventricles, and you can see the locations would be the choroid plexus of the lateral ventricle, choroid plexus of the third ventricle. There's a choroid plexus, let's get rid of that, down here at the fourth ventricle. So CSF is produced in the ventricles at the choroid plexuses, excuse me. It's going to flow through those ventricles, through those pathways that we mentioned. So those little black dots are the CSF making its way. We can see it can fill the subarachnoid space. It doesn't show it going down the central canal, but it fills the subarachnoid space and then can enter into these arachnoid villi and then enter into the venous system and then back to the heart. And the entire thing is just continual. So pretty good video, kind of summing that up. Okay, so last thing I really want to mention with this chapter is looking at the structure of the spinal cord. So if we look at the spinal cord, we can see it's going to begin at the site of the foramen magnum, which is the large opening that we saw in the occipital bone, and it's going to travel all the way down to the lowermost border of your first lumbar vertebrae. Okay, so that's going to be the flow, or the path, the length of the spinal cord. As we mentioned before, there are two bulges. So you have your cervical enlargement and then your lumbar enlargement. That's where the spinal cord thickens because of some plexus, which is going to be a bundle of nerves. So let's look at a picture right here. Okay, so cervical enlargement is here. We can see a bundle of these nerves kind of branching off at that location. Down here, you can find the lumbar enlargement, and we're going to have a bunch of nerves kind of branching off in that location as well. Cauda equina, that's the horse's tail where everything frays. Conus medullaris, that's the cone-shaped distal end of the spinal cord around the first lumbar vertebrae. Filum terminale is that final distal piece of pia mater from around the spinal cord that blends in with the periosteum of the coccyx. And of course, it's going to begin up at the foramen magnum. And then this little bit just talks about the different parts of a spinal cord section. So remember, in the last lecture, I told you that you are going to need to know what side of the spinal cord is anterior and what side of the spinal cord is posterior. So anterior side has a median fissure. Posterior side has a median sulcus. A fissure is deep and wide compared to a sulcus. So the anterior side, you see a very deep and wide groove. On the posterior side, a sulcus is not so deep and not so wide. So you could look at a picture like this one right here, and you can see the difference. On the anterior side, which is up here on this picture, again, access your compass rosette to see that. Anterior side, deep, wide groove, known as a fissure. Posterior side, just a thin little sulcus. Okay, so a thin little sulcus on the back, which is posterior side. So you can always tell which side is the front and which side is the back. Okay, so this picture right here, yet again, just highlighting what side is anterior, what side is posterior. Now, there's a little review from the last chapter. We can see a spinal nerve is mixed, okay, because we're going to have both types of fibers. Okay, so again, information, you have to know if it comes in through the back or comes in through the front. It's going to come in through the back. So here we see our blue motor or our blue sensory afferent nerve fibers are coming in through the back. Okay, so sensory in through the back. Here's a little interneuron. And then motor or outgoing or efferent, these efferent ones come out through the front. Okay, so again, in through the back, out through the front. Here we can see they're together, making your two-way traffic of all spinal nerves are always two-way traffic. This happens to be a 3-neuron ipsilateral reflex arc because it's the same side of the body. It's all from the right side of the body down here, sensory, and then it goes to the right side of the body, motor. We can see once these fibers do split from the spinal nerve, all the cell bodies are clustered together, and this dorsal root ganglion is a swelling, it's a thickness. So you can always kind of tell the backside because these sensory neurons always have this swelling, this ganglia. You can see it right here as well. Just another clue that this is the backside. Anterior side, you don't have that thick swelling. So here's that backside. And then you have, from splitting of the spinal nerve, you have the posterior nerve root. And it actually looks like roots in the ground. They're little roots, like little fingers that physically attach to the spinal cord. And that's where all these nerve fibers pass through. And then on the anterior side, when information comes out, outgoing motor efferent, you still have your anterior nerve root, all these little like fingers. And then the fibers make up the two-way traffic again with the spinal nerve. Gray matter for the spinal cord is kind of the centermost region of the spinal cord. It kind of looks like a capital H, okay, where we have different columns, like this is a lateral column on the sides. Anterior column is up here in the front. This is the anterior side. The posterior column is back here. And then as far as the medial area, this little crossover is called the gray commissure. So you can see right here, gray commissure is just this center part. And then of course there's that hole running through the middle, which is a central canal filled with CSF. And then your white columns, you have the anterior white, the lateral white, and then the posterior white back here. Okay, and the white columns have this strange name, funiculi. We just typically refer to them as white columns. Okay, so that is a structure that you see associated with a segment of spinal cord. And then the remainder of the chapter, we are looking at regions of the brain, and this we will discuss when we do our brain dissection together in class.